So I must say, after the whole salmonid session, here I am talking about common carp. However, I must disclaim there is a similar analysis ongoing for the salmon as we speak, so it can be also generalized. But what will I be talking is about promoters and transcription initiation in common carp. So why do we bother? So why the promoter important? The transcription starts from the promoter, and the promoter takes input from all the regulatory regions in the genome to drive the gene expression, timely gene expression. And the promoter is composed by several elements, which some are known, some are not known yet, some are yet to discover. And in general, we have like a couple of different classes of promoters. Like we have sharp promoters, which have like one sharp initiator, like downstream of around 35 base pairs from the Tata box. We have broad promoters, which are characterized by CPG islands and with multiple transcription start site. Those are mostly like housekeeping genes, but we have the most interesting one is the developmental promoter, which has like these thick CPG islands, which extends deep into the gene body, and they are responsive to long-range regulation, from which like they can span several megabases away. So why is a common carp actually a good system to analyze that? First of all, it's closely related to zebrafish, which is like one of the most important model organism in general, and there was a lot of discoveries and a lot of analysis on promoters happening in zebrafish. So we can actually compare the early development of carp with the zebrafish. But unlike zebrafish, it had a whole genome evolution, just like salmon did. However, it's not the same as in salmonids. Salmonids has autoploidization, while the cyprinids had like allotetraploidization, which means their genome was derived from merging two closely related species. So what's important is that we have like one system, one controlled system, which has like two subgenomes inside, and we don't need to control like for any outside environmental inputs to see the to account for the difference. So how we did the analysis, we used the cap analysis of gene expression or cage, which is currently the most unbiased method to analyze promoters on a single base resolution. We biotinylate the 5' cap, and then we do the pull down, we construct the library, and like the beginning of the read is actually like where our initiation start, and it gives us also like a quantitative method because the intensity of the peak is actually the amount of expression. We can also cluster this initiation to get the promoter region, or how we call it, tech clusters. I will show some data later on. So how did the data quality look like? So as I said, we were interested in zygotic genome activation, which where like before the zygotic genome activation, like maternal transcripts are dominated, and after the zygotic genome activation, the zygote starts to express their own genes. And when we did the PCA of the samples, we actually got a nice developmental trajectory with the pre-zygotic genome stage like being on the left-hand side, and everything else like following a nice developmental trajectory on the right-hand side. And when we look at the specific denucleotides from which the transcription starts, because the transcription doesn't start from random nucleotide sequences, it prefers pyrimid and purine denucleotides, or pyridine and cytosine. We can see that all of our samples are indeed enriched in pyridine purine initiators, although we can also see some other interesting dynamics, some of them which is known, some of them which is new. I won't go into detail because it's still an ongoing analysis, but what we can say from this is that we have a high quality data set, and that we have some novel observation which will give us some further biological insights. In terms of tag clusters, so when we do the clustering of individual transcription start sites which are close together, we can see that most of the promoters actually, which was quite surprising, were active in all four analyzed stages. But other subgroups involve zygotic promoters, which are active in zygotics after the zygotic genome activation, and we have like one large group which is maternal specific promoters. We have also other groups. I won't go into details with that, but one most important aspect is that like 80% of our tag clusters for all samples are actually found in the ensemble annotated TSS region, like plus minus 3 kilobases away, which is like a good sign that we have like that the annotations broadly overlap, not perfectly, but broadly, which I will tell in a second as well. As I mentioned, we have like sharp promoters and broad promoters. So like the interquantile width of the promoter is one of the measure of quality which we also consider. So we have this peak for sharp promoters, but we also have this other distribution of lengths for the broad promoters as well. And what we can see is basically that the maternal promoters are in general sharper than those in zygotic stages, which is something which we would expect from the results we already had so far. And I will tell just in a second, just to show you one example. So in the genome browser, how a sharp promoter looks like. So we have like a canonical CA sharp initiator, which is located downstream. Okay, so this is an antisense direction. So here is the Tata box, here is the sharp promoter. And on the other hand, like the broad promoter has multiple sites spread over a larger region. So let me go back into why the maternal are sharper than the zygotics. So we have my group has discovered, actually before my time, that there are two grammars coordinating on the same promoter between the maternal transcript and the zygotic transcript. Well, the maternal transcript, and then there is this shift in transcriptional start site between the maternal stage and zygotic stage, because the maternal transcripts are characterized by the sharper promoter located around 30 base pair from the W box. So the W box is the 80-reach oligonucleotide sequence, similar to Tata box, but a little bit degenerated. While the zygotic promoters are defined by the nucleosome positionings signet, and they span a broader region compared to the W box dependent start site. So, and just to show how this looked in zebrafish. So if you look, each of the line in the heat map represent one promoter, and when they are when they are centered on the maternal dominant TSS, we can see this W box here. But if we center them on the zygotic TSS, you can see that we have a nucleosome positioning signal really sharp here, and that the W box doesn't influence actually too much the position. So these are the this is all already published in zebrafish. However, we perfectly recapitulated these data in our carp samples and looked practically amazing. The only question which we now have is, so we now know, even we had a whole genome duplication, that both of these grammar are found still on the same promoter, although we had like a whole genome duplication, meaning that a lot of them didn't lost one of the grammar. So, however, we still don't know if some of them basically did lose the grammar, and how much these mutations in this sequence are tolerated to change the expression and lead to subfunctionalization of the gene. So here is one example from the genome browser showing like the maternal W box driven initiation and the shift happening in the after zygotic genome duplication. So let me tell just one segment about how actually our transcriptional start site correlates with ensemble annotated start of transcription. As we can see, like around only 30% of the annotated promoter are actually overlapping the precise TSS in ensemble, while majority of them actually are further away. And what we can see also that they are mostly found downstream of the ensemble annotated TSSs, meaning that actually ensemble tends to overestimate, actually RNA-seq data in general tend to overestimate the transcription start site, taking like the longest, probably taking the longest variant of the transcript. And here is one example from the genome browser where you can see that the annotated TSS is here, while downstream we have the actual promoter and the actual transcription start. So just to finish, like something basically biologically meaningful for the carp, since we have this whole genome duplication, what we can actually look is the difference in the initiator sequence between the two subgenomes. And so what I did next, I counted the mutations between the two subgenomes. So here is the initiator in the subgenome 1, and here are the initiator in subgenome 2. And what we can see basically that most of the changes between the two occur from the canonical initiator to another canonical initiator, meaning that they want practically to maintain their expression level, and there is a pressure on maintaining the canonical initiator. The question is what happens if we have a canonical initiator transitioning into non-canonical initiator, or the other way around. What we see basically is that the transcription, if we have a canonical to a non-canonical transition, that the expression of that particular transcriptional start site drops, while if we have like a transition from a non-canonical initiator to a canonical initiator, the expression rises, which means that there is like first an evolutionary pressure, and it might be a source of subfunctionalization as well. And here is actually one example of a non-canonical initiator transitioning into a canonical initiator, and with we see that the expression actually increases 4 times. So with that, I would stop for now. We have some summaries like CARP indeed represent like a good system to look at the promoter evolution and expression initiation. Cage is an adequate method to do that, and this data set actually presents like an opportunity to tap into promoter evolution. And with that, I would like to thank my old group at Imperial College London, also my new division in my new institute, Ruđer Bošković in Zagreb, where I recently started a new tenure track position with the hope to establish my own group, and that would be all for me for now.